Thanks to Sarah and Tammy for inviting me out here. As Kent mentioned, I'm gonna focus primarily on a potpourri of topics, really around how do we maximize different ways to maximize information coming out of brain imaging studies. So I'll go through these topics, but I'll kind of just jump right in and start with the concept of big data. So big data is kind of a buzzword that's out there now. Everybody's doing it, and but just in terms of motivation from a brain imaging study, okay, well we have a brain disorder, so we should be using brain imaging to study that. So let's go big. Let's collect more data, image at the best resolution possible, lots of information. But then we become this poor guy buried under a pile of data. I've got to use the triple picture. That was excellent, Kent. So that's going to go here in my next talk. So ideally what we want, though, is we want methods that can try to help us extract meaning from the data. Basically, there's a lot of data, very high dimensional. Can we pull out some of the essence that's going to be useful to fit into all these models that we're trying to use? And one more slide on this, just to kind of talk about simplicity and complexity. In the context of a classifier, for example, it's valuable if it can discriminate what we care about, sensitive, specific, and we can replicate it. Robustness, not necessarily of value by itself. We just need it to be robust enough, the biomarker needs to be robust enough to replicate prediction across samples. So just one example here, I've got sort of increasing complexity going from left to right, average over the whole brain, just a frequency distribution, and a seed-based connectivity map. If we want to predict age, for example, we can do really well with the mean over the whole brain of resting fMRI study or a structural scan. We don't need this extra complexity necessarily to help us, but if we're looking at something like schizophrenia, for example, that's not quite enough, and we can get better performance if we incorporate these more complex measures like seed-based or other kinds of things. And so we shouldn't necessarily avoid these more complex approaches just because of greater potential statistical noise or more processing steps. Often hear Occam's razor used to shorten the reins on otherwise unbridled enthusiasm for complexity. So let's not get too complex. Let's keep the methods as simple as we can. We need to temper that with Carl Menger's law against miserliness, which says entities must not be reduced to the point of inadequacy. Okay, so we need something that is going to be complex enough to capture what we care about. Ultimately, replication, as we all know, that's the gold standard here. And the most, if we simply do validation and out-of-sample prediction, the most suitable classifiers will emerge naturally, regardless of their complexity. So, that being said, obviously I'll focus a lot on connectivity. So that's been one of the things that's really sort of taken off with the Human Connectome Project and lots of other things. This is a typical kind of analysis you'll see in those studies where you have, this is a group ICA, independent component analysis, which basically gives us, in resting fMRI data, a bunch of different brain networks here that are coherent in time with themselves. And they're grouped into domains here, so anatomically. So here are visual areas, here's so-called default mode network areas. And in addition to this, the within-network connectivity, we can take the time information in each network and look at how these networks interact with one another. So that's what this is kind of showing us. You'll see lots of matrices and different ways of displaying things like this. But for example, you often see this kind of a blocked structure. This is the cross-correlation among these different networks. Okay? So here's the visual networks right here. And you'll see this block here, which shows that they're sort of talking to themselves more strongly than to the other networks. Here's the default mode network, and it's also talking to itself, but weakly anti-correlated with these other things. So, so that's sort of the among network connectivity. And then we also get information about how things are fluctuating in time. Are they fluctuating fast or slow? And that's kind of the frequency distribution. So, obviously this is a little bit scary if you're focused on a particular set, you've got a very specific hypothesis about one particular brain network or region or structure. For example, as I think the previous speakers kind of alluded to, this is some data from one of Kent Hutchison's studies, where we basically looked at the network connectivity among a variety of networks, and these are the ones that sort of emerged as showing differences between substance users. basically smokers and drinkers. And so it's really, I think the first speaker was talking about, you know, the interaction, right, with this core network that might be dysfunctional. Well, the interaction is important as well, right, among all these different networks. And we're seeing basically, in this context, a hypoconnectivity pattern in both alcohol and nicotine use, ranging from frontal, temporal, cerebellar, insula, and thalamus. Okay, so there's a lot of networks here that are implicated, and to just ignore those, I think, is going to miss some of the important information. And I'll touch back on that later. But you'll see I'll be going down and down levels and levels here. We can still get more information from our data, right? So there's been a lot of emphasis recently on looking at dynamics in connectivity, or the chronic-tomics. So, for example, if I have two regions here that are connected, well, maybe they're not just connected on average. Maybe that's missing a part of the story here, which in this case we have a strong coupling at the beginning and a weak coupling at the second half. And if we just look at the average, we miss that. And you can extend existing connectivity methods pretty easily to capture this information. So for example, instead of calculating the cross correlation among the whole time course, we can use some sort of a windowed approach or adaptive approach to basically get a time series of these matrices. Well, now what do? Well, you can just do something, basically cluster these down into a small number of recurring connectivity patterns that subjects go into and out of over time. And then that lets us do things like, for one subject, we can say which of these states were they in at each point in time. So this subject went from state 3 to 2 to 4, etc. So ultimately, we're trying to get, some smaller dimensional things that we can use in our models while still respecting the complexity of the data. And just to kind of give you one example of how this is used, this is some data we got from Enzo Tagliazucci, where they have concurrent EEG fMRI, and they did sleep staging with the EEG data. So you'll see here, state 1 maps fairly uniquely onto the wake condition, and state 5 here, which loses these anti-correlated regions, is mapping fairly uniquely onto the deepest sleep stage, N3. So unfortunately, when we tell our subjects to rest, they actually rest. So, and they'll tend to get drowsy and fall asleep, and it's an important confound that we need to actually take into account. And he showed in another paper that you can, if you look at the functional connectivity 1000, there's like 4,000 data sets now. Pretty much all of them get pretty drowsy about halfway through 5 minutes, and a substantial number of them fall asleep by the end of the study, or get into at least a deep sleep state. So we have to be careful we're not just classifying sleep, for example. This again is some of Kent Hutchison's data, where we're using a dynamic analysis. And so here we have 460 subjects. I know I'm going really quick here, but these are all the different networks, and we get, basically they're grouped into different domains like before, where we have subcortical regions, we have visual networks, etc. And then these are 5 different connectivity patterns that were extracted using that dynamic analysis I mentioned before from a 5-minute resting scan. And then when we looked at one parameter, which is how long, given what substance they are, so we have controls, alcohol only, smokers, and combinations of substances, etc. How long are they spending in each of these states? And we see that that 2 of the states here are showing strong differences between the substance users and the controls. And if we look a little more carefully at that, we'll see these are the same 2 states I showed you before in terms of the amount of time they're spending. This is a state that controls spend most of their time in this state here. And the substance users are actually moving from this state over to this state. And in particular, you're losing this, you'll see this default mode network coupling with actually a lot of different networks, including the subcortical networks. And so this is a kind of analysis that if you're not looking at these kinds of dynamics, it's getting all blurred and averaged together over the 5 minutes. We can go another level down. We can say, well, maybe I don't want to assume that somebody's just in one state at one time. Maybe I want to think about connectivity as sort of a state space. So consider this, we have state 1 versus state 2, and there are different degrees, sort of how strongly am I represented in one state versus another. And so here's two subjects in red and blue, kind of at different time points, they're kind of moving around in that state space, for example. And I can characterize that with these sort of numbers here. So for example, this is the first time point, and this is state 1, and this is state 2, and it tells me the level that they're in, basically the coordinate on that grid system. And so this we call a metastate, just because we wanted to come up with another terminology and add more jargon to the field. But given the metastate, what this does is it captures the dynamics, and then we can characterize this in some very simple parameters. Things like the number of distinct metastates that they entered, the number of times they changed, the total distance they move around in that state space. This is some data from a large schizophrenia study, and regardless of which approach we use to estimate the states, you can see all of these numbers are negative. We're basically getting consistently reduced dynamism in the schizophrenia patients. One interesting parameter here is something we're calling a hub state, which is basically a state that subjects appear to get into, and when they get into it, they tend to stay there longer. It's almost, think of it like a sticky state, so to speak. And schizophrenia patients in particular have significantly more of these hub states than the healthy controls, and this correlates with negative symptoms as well in schizophrenia. So again, getting down to some parameters that we can sort of understand or at least have some intuition about, but still being able to capture and use all of the data. And here's one more just to kind of as a teaser. But suppose we've got the connectivity pattern here, how these regions are interacting, how strongly they're interacting. But maybe we also want to capture the distribution in the frequency domain. For example, here's two different states here. This one has a typical bold-related frequency distribution. This is low frequency to high frequency. And this one here is much lower in frequency. And then we also have a phase information that's captured, kind of the delay, or the delay in the interaction between these different things. And we can do this with something called a time frequency analysis, building on the work of Katie Chang and others, and separate different states that look completely overlapping if you only look at the correlation or the amplitude. Okay? So let's talk a little bit about classification. So obviously that's if we want to do an individual subject, and that's of use in these kinds of trials we're talking about, we want to say something about an individual. We're not necessarily only interested in a statistical difference between a large group of subjects. So as either Yogi Berra or Niels Bohr or Mark Twain said, prediction is difficult, especially about the future. If someone knows where this quote came from, please let me know. I haven't been able to sort it out using Google. So one thing we were interested in was whether other groups could sort of do what we think we're able to do with classification. So this is a schizophrenia data set where we basically calculated in an unbiased way connectivity matrices and from structure and function. And we threw it up on this website called Kaggle. And it's a competition website where you have data that you can see and data that you can't see, and you compete, and et cetera. And surprisingly, we got over 2,200 submissions to this competition. Each dot here is a submission. And the majority of the performance was above these red lines, which is 80%, and many of it's close to 90%. There's clearly value in this data in terms of differentiating. Except for these poor groups. I don't know if they did really, really, really bad, but you'll see that the bulk of people actually did quite well. And interestingly, then we can take this and we can look, well, let's see, can we somehow leverage the best groups? And, there's a bunch of classifiers. Say we take the top 10 and we say, let's use a weighted voting. So if there's more than 6 that voted A versus B, then we count it as A. And that actually works all the way up to the top 65 classifiers. So we're clearly still not fully leveraging all the information in this data, and we can learn from one another about that. This kind of approach works. We've looked at a lot of different data sets. This, for example, is a mild traumatic brain injury data set, where in this case, we have the two connectivity matrices here for the different groups. These are the controls, and you'll see that in the traumatic brain injury folks, there's an increase. There's a hyperconnectivity among those regions. And if we use this for classification, there's been a lot of papers primarily focused on diffusion, because you might imagine that sort of the mechanism makes sense. The brain is moving and twisting, et cetera. But the resting fMRI actually does apparently better in a cross-validated classification. We're getting close to 80%. And if we then look at which features in the data, which networks were the most informative about the classification, it's these long-range connectivities between cerebellum and cortical regions that are actually showing the most informative information there. Getting to the dynamic versus static in terms of classification, what we found in several different data sets, several different domains, is that the dynamic information is giving us better performance when we try to do individual subject classification. We can do really well in a lot of different cases with the so-called static connectivity. So this is a 3-class problem. We have schizophrenia, bipolar, healthy controls. And this again is some of Godfrey Perlson's data. And so we're looking at, the static FNC does respectively quite well above chance. But we get a significant bump when we incorporate the dynamic connectivity information into the problem. And this also increases our performance when we look back at that mild traumatic brain injury. And if we look at some other data sets, we've seen this sort of consistently. So it looks like it might be something we should typically use for that, although we're still looking at it. Okay, so my son hates this picture, but this is Jakin doing data fusion. He's 12 now, so anyway, I think multimodal imaging, complementary information we all want to do. It's hard to do. Just to kind of motivate, I like to show this slide from a number of years ago, where let's just think about, here we've got 2 different tasks. They were collected separately. One is an auditory oddball task, one is a Sternberg task. And if we just look over here, these are individual subject sort of joint histograms of task A versus task B. And you'll notice that in the case of the schizophrenia patients, you see this angle to the distribution, whereas in the controls it's more flat. What does that mean? Well, it's telling us that the activity patterns for these very different tasks are less unique in the patients. They're sort of activating their brains in a much more similar way. So that gets at issues of, is it a cognitive efficiency thing or is it something else? This kind of information you cannot see if you don't look at 2 different domains, right? And this is true also of diffusion and structure. A joint analysis can capture that sort of thing, but you can't get it with 2 separate analyses side by side. Not that those aren't valuable, but they're missing that. And we can also use this to look at other variables. So we have lots of assessments and questionnaires, the clinical information. In this case, we were just simply predicting a cognitive battery called the matrix battery using a combination of functional and structural data. That's not for me, right?
Nope.
Okay, good. Get off, get out. I was told Sarah was going to be very strict with time. So, okay. So here's our prediction. You can see the controls generally do better on the battery than the patients, but we're getting upwards of 80% ability to predict. And where this gets even more interesting is when we try to intersect the domains of cognition to these different networks that we're extracting. And then we can turn that around and say, well, given these networks that we've implicated, what does it tell us? The degree to which these different networks are correlated to these domains. It helps us sort of interpret and understand what's going on. And just one slide here to say, we've got all of you probably that do imaging have multiple kinds of data, and pooling it together can help us. So we have fMRI, diffusion, and structure. This is a joint analysis that pulled out covariation that's similar among all 3 modalities together that was differentiating the schizophrenia patients from the controls. Okay? And this is some data from the BSNP study that Godfrey Perlson collected. And what they did was look at using a parallel ICA algorithm they were basically an extension of data fusion, is to take this genetic data that we have a lot of and this imaging data that we have a lot of and try to put it together. And so you can pull out covariant networks of, in this case, it was multiple subnodes of the default mode network and genetic networks. And then they did an ontology analysis, basically looking at the terms that were represented in the pathway analyses. And there was an overrepresentation of psychosis, for example, suggesting that these gene networks mediate the risk of psychosis via these default mode networks in the probands. Okay, so let's see if I can get through the last slides real quick. I've got I wanted to kind of say, there's even more complex things we can do. So not just to do complex things, but deep learning has become very widely used in things in companies like Google and Facebook and Amazon. We were interested, and basically what you're doing is you're taking a model like an ICA model and stacking it on top of another model. Okay, so the idea is to capture nonlinear things and very subtle things. You need lots of data to train these models. And so our question was, well, are these something that can be useful in imaging data? And it looks like initially that the answer is yes. So this is going increasing the depth from 0, 1, 2, and 3. And basically you see that we get an increased separation between two groups, both in the training data and the testing data in this study. And here's another example from a Huntington's disease study. We have 4,000 scans here. The algorithm knew, this is structural data, and the algorithm knew nothing about disease severity. But we can characterize disease severity through CAG repeats, and basically color-coded the results on this plot from least severe to most severe. And you see this gradient emerged that the algorithm didn't know about, so it naturally found that pattern, which was quite striking to us and encouraging. And it's sort of we're starting to look at multiple ways to look at this. But if we can find those patterns in our data, we can go back and look at which brain networks, which features are relevant for it. And we're also looking, using these kinds of models, this again, I'm just sort of at a high level, you can use deep learning to do translation between, so Google's very interested in, you see the translate button on there. They're trying to translate, for example, between English and French by training their models on these large corpuses of text. And we can actually use that to translate between multimodal brain imaging data. So we can look at, for example, structure and function. We're actually using these ICA networks to get to something which we're calling sort of a word in structure and function. And I'm kind of glossing over the details. But it gives us some really interesting ways to kind of look at the relationship between these very different domains. So last couple slides. Hopefully I got a couple minutes, yes. Sorry. So just briefly, working with lots of data, studies are getting bigger and groups are pooling data together. And so management systems are important for doing this in a way that can not only capture just the imaging data, but all the clinical data and other things. This is a tool that we developed called Coins, which has been used to manage quite a few different studies around the world. And you can see here data providers and data consumers are quite well represented. And I just to kind of mention the importance of trying to better integrate all these different databases we're setting up, instead of just creating a sort of assessment database and an imaging database and a genetics database. They need to be better integrated. You've probably all gotten 5 spreadsheets from different sources of data, and it's a lot of work to figure out how to get those together. And you also get better data because you can, for each of these things, you can monitor the data as it comes in and track it. And just some examples of that from the imaging domain. This also works for questionnaires and assessments. But this is a data, this is, I think, something like 3,000 or 4,000 imaging data sets, where we took resting data and we calculated some quality control parameters from it. And then we just projected it onto this 2-dimensional plane. So things that are, dots that are close together are kind of similar. You can kind of think about it that way. And then the structure actually looks useful. So for example, this big cluster here and this big cluster here both had, they were good data sets, but they had higher motion in the resting fMRI data, for example. This particular cluster all the way out here was where some of the automated processing failed and the brains were normalized upside down or backwards or something like that. So it gives us kind of a real high-level view. One more slide after this, and I'll be done. Okay. And we can also look at measures of interest. So this is basically taking volumetric data from a free surfer study. And again, we have multiple scanners represented here, and we just color-coded it by gender, males versus females. And you can see there's actually a difference in brain structure. This is not overall brain volume. That's been normalized out here. But we have more males represented here, more females here. And we can look at the raw data as well, or not quite raw, but all the voxel information. This is a gray matter data set. There's 10,000 scans here from 2 different scanners, and then we color-coded it by age. And so these folks up here, we range from 18 months, which is this little bump sticking out here on the left, and all the way to almost 90 years old. And obviously, as I mentioned, age is a great predictor. Imaging data is a great predictor of age. It's represented in this plot. That's our data sharing portal. And I'll skip that slide because I've run out of time. And we try to make our analysis tools available to the community so that it can be used and hopefully made better. And obviously, I don't always wear the same shirt, but I think I didn't plan that. But anyway, this is my lab, and obviously there are lots of other collaborators involved in this, and I'm thankful for all of that, and without which the work wouldn't have been done. So thank you.